Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Hypno and its relationship with Nidoqueen and Nidoking, as they are all part of the Ceratomorpha clade. Ready? Let's go! Ceratomorpha are a suborder of the odd-toed ungulates, Perissodactyla, which today include the rhinoceroses, Rhinocerotidae, and tapirs, Tapiridae. They represent an evolutionarily significant group, distinguished by characteristic skeletal features and a long fossil record. The origins of the Ceratomorpha trace back to the early Eocene, around 55 million years ago. Fossil discoveries such as Homo gallix and Lophiaspis moretii are considered early representatives of this lineage. The evolution of Ceratomorpha paralleled the radiation of other odd-toed ungulates, such as the horse-like Hippomorpha. Within just a few million years, the tapir and rhinoceros lineages began to diverge, developing distinctive features like toe number and tooth structure. Fossil species, including the extinct Brontotheres and the primitive Homo gallix, illustrate the past diversity of this group. A shared feature among Ceratomorpha is the development of strongly pronounced transverse ridges on the upper molars, an indication of their specialized plant-based diet. Tapirs possess 4 toes on their front feet and 3 on their hind feet, a trait they share with early forms like Homo gallix. Rhinoceroses, on the other hand, have reduced their digits to 3 toes per limb. The skull structure in both groups is massive, with rhinoceroses particularly noted for their distinctive horns, which, interestingly, are made of keratin rather than bone. Tapirs, Tapirus, are the only extant genus of the mammalian family Tapiridae within the order of odd-toed ungulates, Perissodactyla. Once a highly diverse group, the genus today comprises 4 living species. These animals have a robust body and a characteristic short trunk. They primarily inhabit dense tropical forests and feed mostly on soft plant matter. Tapirs are considered an ancient genus, with fossil evidence dating back to the Middle Miocene about 14 million years ago. Today, tapirs are found in South America, lowland and mountain tapirs, Central America, the Central American tapir, and Southeast Asia, the Malayan or Shabrakan tapir. Although tapirs vaguely resemble pigs, their closest relatives are actually horses and rhinoceroses. They range in body length from over 100 to 250 centimeters, with a short tail measuring 5 to 13 centimeters, and stand 73 to 120 centimeters at the shoulder. Adults weigh between 110 and 320 kilograms, the largest living species being the Malayan tapir, Tapirus indicus. The fossil species Tapirus augustus, also known as the megatapir, was significantly larger, exceeding the Malayan tapir's size by 25% in all measurements. On the other end of the spectrum, small extinct species like Tapirus polkensis weighed only 110 to 140 kilograms. Tapirs have a bulky, somewhat clumsy-looking body that narrows at the front and is rounded at the rear, an adaptation that helps them move through dense forest undergrowth. The American species are typically brownish-gray, while the Southeast Asian Malayan tapir stands out with its striking black and white coloration. The tapir's head appears relatively small compared to its body. Its eyes are small, while the ears are oval, upright, and highly mobile. In some species, the ear tips are white. A defining feature is the trunk, formed from the upper lip and nose. The legs are relatively short and slender, and like all odd-toed ungulates, the main axis of weight-bearing runs through the third toe, which is also the largest. Each front foot has 4 toes, with the 3 pointing forward being the most developed, while the outer one is reduced in length. The hind feet have 3 toes. The tapir's skull is generally elongated and flat. Among South American species, a sagittal crest, formed from the inner ends of the parietal bones, runs along the midline of the skull roof. The Central American tapir, Tapirus bairdii, and the Malayan tapir, Tapirus indicus, do not have this crest. Instead, they feature a pair of bony ridges, parasagittal crests, on the parietal bones near the brain case, separated by a narrow, flat surface. The occipital bone is rather short and angular, and the nasal bone is small and weakly developed. Typically, it lies well behind and above the premaxilla and does not connect to it, resulting in a large nasal cavity. The entire front of the face is greatly reduced to accommodate the complex musculature of the trunk. The taper's dentition is barely reduced, resembling that of early mammals. Adults have 42 to 44 teeth, following this dental formula. The incisors are small and conical, except for the upper third incisor, I3, which is significantly enlarged. The canines are also conical, very large in the lower jaw and much smaller in the upper. Together, the lower canine and the outer upper incisor form an effective biting tool. A diastema, tooth gap, separates the front teeth from the cheek teeth. The premolars resemble molars in shape, they are clearly molarized. As in all odd-toed ungulates, the cheek teeth are bilophodont, characterized by two transverse enamel ridges on their chewing surfaces. They are low-crowned and contain relatively little cement, making them suitable for consuming soft plant matter. Fossil tapir species share these features, though their premolars vary in how molarized they are. The nose and upper lip fuse into a small prehensile trunk used to find and grasp leafy food. The Malayan tapir has the longest trunk. The lowland tapir, Tapirus terrestris, has the shortest. Since fossil tapirs show similar skull structures, the trunk is considered typical of the genus. Like in elephants, the tapir's trunk is a fully muscular tube with two open nasal passages, but it is much shorter. It lacks any internal bone structure, and its connection to the facial skeleton is a distinct evolutionary adaptation, differing sharply from other odd-toed ungulates. The high mobility of the trunk is enabled by three major muscle groups that run lengthwise, crosswise, and in spirals. Several facial muscles, such as the musculus levator labii superioris and musculus levator nasolabialis, have undergone significant changes to support this flexibility. Unlike elephants, however, tapirs did not develop further cranial or dental changes. The trunk is shorter and less versatile than an elephant's, limiting the size of objects it can handle. Nevertheless, tapirs, like elephants, use their trunks to forage, manipulate food into their mouths, and for smelling or even snorkeling. Therefore, unlike trunk-like formations in animals such as pigs, elephant shrews, or dik-diks, the tapir's trunk qualifies as a true proboscis. As with all odd-toed ungulates, tapirs are hindgut fermenters. Most digestion occurs in the rear part of the gut with help from numerous microorganisms. They have a single-chambered, relatively small stomach. Their intestinal tract can reach up to 11 meters in length, though the cecum, blind gut, is relatively small for this group. Each kidney contains about 3 million nephrons and weighs up to 390 grams, together making up only about 0.5% of body mass. Tapirs today have a split distribution. 3 species are found in Central and South America, ranging from southern Mexico to southern Brazil and northern Argentina. The 4th species, the Malayan tapir, lives in Southeast Asia, from Myanmar to the Malay Peninsula and on Sumatra. This divided distribution is a remnant of a once much broader range. During the Miocene and Pliocene, tapirs inhabited much of the Eurasian continent, excluding the Indian subcontinent, as well as large parts of North America. South America was only colonized in the mid-Pliocene, following the formation of the Isthmus of Panama and the subsequent Great American Biotic Interchange. As climates cooled and seasons became more distinct, accompanied by the spread of open landscapes from the Miocene through the Pliocene and into the Pleistocene, tapirs disappeared from Europe, Northern Asia, and North America. Today, their habitats include forests, primarily tropical rainforests, but also cloud forests in the mountains. They rely on access to water and can be found from sea level up to elevations of 4,500 meters. Because tapirs are a conservative genus with minimal physical changes over time, this is believed to apply to fossil species as well. Tapirs are solitary and territorial. When individuals encounter one another, they often behave aggressively. Males and females come together only briefly during mating season. Territories range from 1 to 8 square kilometers, with females sometimes occupying larger areas. Territories usually contain multiple sleeping, feeding, and wallowing spots. Paths and boundaries are marked with feces and urine. Tapirs are nocturnal and retreat into dense undergrowth during the day. At night, they forage, moving forward with their trunk held close to the ground. they often remain near water, where they are excellent swimmers and divers. Mud baths are commonly observed. Generally, tapirs are shy and cautious. When threatened, they flee, often into water, but will bite if cornered. Their hearing and sense of smell are well developed. Tapirs are herbivores that mainly consume soft vegetation. In addition to leaves, they eat aquatic plants, buds, twigs, and fruits. Their long, muscular, flexible tongues allow them to reach leaves on thorny plants. Several hundred plant species are known to make up the diets of various tapir species. As they move through the forest, they spread plant seeds through their feces, making them an important ecological contributor in tropical forests. To neutralize toxins sometimes ingested with their plant diet and to maintain nutrient balance, some tapirs regularly visit mineral and salt licks. Tapirs are also highly dependent on water and adjust their drinking habits to local conditions, consuming significantly more water in dry areas. Tapirs, Tapirus, make up the only living genus within the family Tapiridae, rendering the family monotypic. Their closest living relatives are rhinoceroses, Rhinocerotidae, while horses, Equidae, form a more distant group. Together, all three families make up the order of odd-toed ungulates, Perissodactyla. According to molecular genetic studies, horses split off from the common lineage at the start of the early Eocene, around 56 million years ago. The divergence between tapirs and rhinoceroses occurred later, during the Middle Eocene, approximately 47 million years ago. Broadly speaking, tapirs and rhinoceroses are grouped within Ceratomorpha, which also includes all their extinct relatives, tapirs within the superfamily Tapiroidea, and rhinos within the Rhinocerotoidea. The counterpart group to the Ceratomorpha is the Hippomorpha, which includes horses and their fossil kin. Today's species poor tapir family was once far more diverse in evolutionary history, a trait shared with its close relatives within the superfamily Tapiroidea. Several extinct families are distinguished within Tapiroidea, though their exact relationships remain unclear. Among the most basal are the Hyracoidae, Deparatellidae, and Lophioletidae. While the first group is known from both North America and Eurasia, the latter two are endemic to Asia. All appeared during the Eocene and show strong dental similarities with tapirs. Some Lophioletidae species show skull modifications that may suggest the development of a short trunk. However, these may represent independent evolution, since none of the three families are considered direct ancestors of modern tapirs. Some researchers even place them closer to the Rhinocerotoidea. More closely related are the Heloletidae, another Eocene group from North America and Asia. They are sometimes regarded as the ancestral group from which tapirs evolved. Besides sharing a similar body plan and dental features, including a tendency for premolars to resemble molars, they also show reduction in facial bones, creating space for the complex trunk musculature, much like modern tapirs. A complication, however, is that some Heloletidae species are also classified within the tapir family itself, making the group partially paraphyletic. In addition to the crown genus Tapirus, which includes all living species, the tapir family, Tapiridae, contains several extinct genera. Many early forms from the late Eocene overlap with the Heloletidae, creating classification difficulties. The oldest unquestionable member of the Tapiridae is Protopirus, which first appeared in the Oligocene and was present on both sides of the Atlantic, in North America and Eurasia. The exact dispersal routes of early tapirs are still poorly understood. Early tapirs already closely resembled their modern counterparts in terms of rear dentition and trunk structure, but they differed notably in the front teeth, for example, lacking the enlarged, canine-like upper third incisor found in modern forms. Such traits only appear in more derived Miocene genera like Tapirus, Tapirabus, and Tapiruscus. Today, 4 tapir species exist: the lowland tapir, Tapirus terrestris, and the mountain tapir, Tapirus pinchak, in South America, the Central American tapir, Tapirus bairdii, and the Malayan tapir, Tapirus indicus, in Southeast Asia. The so-called Kabamani tapir, Tapirus kabamani, described in 2013 from the Amazon basin, has been shown by genetic studies to be nested within the lowland tapir and is therefore not recognized as a distinct species. Genetic analyses also suggest that the Malayan tapir was the first to diverge from the tapir lineage about 25 million years ago. The Central American tapir followed approximately 11 million years ago, while the divergence of the two South American species, lowland and mountain tapirs, occurred much later, around 2 million years ago, likely on the South American continent after their ancestor crossed the Isthmus of Panama via the newly formed land bridge. Together with South America's fossil tapirs, these two species form a closely related subgroup, distinct from tapirs of North and Central America. The relationships among Eurasian tapirs remain poorly understood. A genetic study including the Kabamani tapir proposes more recent divergence times, between 12.7 and 5.6 million years ago for the Malayan tapir, between 7.5 and 3.1 million years ago for the Central American species, and between 0.65 and 0.26 million years ago for the South American tapirs. From an evolutionary perspective, tapirs represent a rather ancient family of mammals. An early precursor to tapir-like animals may be found in the genus Hyraceus from the family Hyracoidae, which appeared during the early and Middle Eocene. A complete skeleton dated to about 44 million years ago was recovered from the Messel Pit fossil site in Germany, though remains have also been found in both Europe and North America. Due to its primitive skeletal structure, Hyraceus is placed by some experts either at the base of the superfamily Tapyroidea or alternatively within the Rhinoceratoidea. Other groups, such as the Deparatellidae, with genera like Deparitella, Teleolophus, and Ironolophus, and the Heloletidae, which include Heptodon, Helolides, and Collodon, are considered basal members of Tapyroidea. A noteworthy Eocene genus is Thuleodonta, which possessed a small upper canine and an elongated diastema, traits that anticipated those seen in later tapirs. Described in 2005 from skull and dental remains found in the Margaret Formation of northern Canada, it represents the most northerly known member of the tapir lineage. Roughly 10 million years younger are fossils of Collodon, including well-preserved skulls from late Eocene and early Oligocene deposits in the White River Badlands of South Dakota, as well as jaw fragments from equivalent layers in Nebraska. Collodon plays a key role in understanding the transition to true tapirs. Its skull shows some modifications, such as a greatly expanded nasal cavity reaching up to the orbits, but the dentition retains characteristics typical of more primitive tapiroids. Some researchers regard Collodon as an early tapir due to its more advanced skull traits, while others, citing dental features, classify it within the Heloletidae. A similar debate surrounds Hesperorhizes, which was described in 2006 and initially considered a basal tapir. The genus includes several skull and dental remains dating from the Middle to Late Eocene, found in the Santiago and Mission Valley formations of Southern California. Like Collodon, Hesperorhizes had a deeply set nasal cavity. The nasal bones, better preserved than in Collodon, suggest the presence of a small trunk or at least a highly mobile upper lip. However, its teeth remained quite primitive. Due to uncertainties in assigning older forms, Protopyrus is considered the first clearly identifiable member of the tapir family. This genus is known from both North America and Eurasia and may have appeared as early as the late Eocene in North America. Several skulls and skeletal elements have been recovered from the Brule Formation of the White River Badlands in South Dakota, which dates to the early Oligocene. There, Protopyrus shared its forested habitat with Collodon. Its skull measured only about half the length of those in modern species. The genus was first scientifically named in Europe, based on a lower jaw found in the phosphate beds of Quercy in southwestern France. Additional specimens include a lower jaw from Gamersheim near Ingolstadt in Bavaria, upper jaw fragments from Aragon in northeastern Spain, and material from the Auvergne-Rhône-Alpes region of central France. Protopyrus was a morphologically diverse genus, comprising several species. Characteristic features include relatively long nasal bones, a nasal cavity extending far back, and only partially molarized rear premolars. Additionally, the limbs of these early tapirs were significantly more slender than those of modern tapirs. In Europe, the appearance of Protopyrus is closely linked to the Grande Coupure event, an extinction phase triggered by climate deterioration that led to a major faunal turnover. As the Oligocene progressed, additional genera appeared. In Eurasia, this includes Paradopyrus and Eotaporus. So far, both genera are primarily known from dental remains, although partial skulls have been discovered in Haslan and Wischberg, both located in Switzerland. While some primitive features, such as a likely short trunk and a small third incisor, remained, the molarization of the front premolars had begun. This was likely a response to the cooler climate phases during that time, which pushed these animals into more open woodland environments. A similar evolutionary trend occurred in North America. Since no evidence of intercontinental migrations has been found, this development seems to have happened independently. The late Oligocene tapirs of North America are thus assigned to the genus Myotopyrus. The type specimen, a facial skull, was found in the Goshen Hole region of Wyoming. Later, Nexuotaporus emerged, a genus that displayed a mix of primitive and modern dental traits. The more simplified premolars, compared to Myotopyrus, represent the former, the more complex molars represent the latter. A direct derivation of Nexuotaporus from earlier North American tapirs remains uncertain, and no clear origin from Asia has been established either. The genus appears to have been widely distributed across southern and central North America. Extensive fossil material, including skull and dental fragments as well as foot bones, has been found from the Fleming Formation near the Toledo Bend Reservoir in Texas. A lower jaw was also reported from the Haystack Valley member of the John Day Formation in Oregon. Early tapirs such as Protopyrus and Paradopyrus in Europe, and Myotopyrus and Nexuotaporus in North America, persisted well into the Miocene. The upper molars used to first describe Nexuotaporus come from the Running Water Formation near Agate, Nebraska, which dates to the early Miocene. Around the same time, the first modern members of the tapir family appeared with Tapiravis, a genus noticeably smaller than today's tapirs but with largely similar dental features, though it had lower crown teeth. The genus was introduced in 1877 by Othniel Charles Marsh, based on an upper premolar from the Kirkwood Formation in New Jersey. Additional finds have been recorded in Maryland. Between the early and middle Miocene, around 18 to 14 million years ago, tapirs nearly vanished from the fossil record in North America and Europe. This period is commonly referred to as the tapir vacuum, which coincided with a climatically favorable interval. In contrast, East Asia retained Plesiotopyrus as the only known form during this gap. The most significant remains come from the Shanwang Formation in eastern China's Shandong province and include a skull and postcranial elements. Additional fossils, like a lower jaw, have been recovered from the Hiramaki Formation in central Honshu, Japan. Plesiotopyrus was somewhat smaller than modern tapirs and showed a mosaic of ancestral and modern traits. Among the primitive features was the absence of a canine-like upper rear incisor, while the molar region resembled that of extant species. Because of this blend, it is sometimes considered a side branch of the tapir lineage. By the beginning of the late Miocene, around 10 million years ago, the very small Tapiriscus appeared in Europe. The oldest known fossils are isolated teeth from Melchingen in Germany's Swabian Jura. A much larger and more diverse set of remains came from Dorndurkheim in Rhineland-Palatinate, including nearly 200 isolated teeth, jaw fragments, and some foot bones. The type specimen, consisting of 4 isolated teeth, was discovered in Szakvar, Hungary, and introduced by Miklos Kretzoy in 1951. The genus Tapirus first appeared in Europe during the Middle Miocene, about 14 million years ago. Its direct ancestor is unknown, though Protopyrus is a candidate. However, since early Miocene fossils of Tapirus are lacking in western Eurasia, the genus likely migrated in from Asia. While older Tapirus finds were once reported from the Oligocene, they are now considered dubious and often reclassified. Several species evolved in Europe. The earliest, T. telleri, is known from only a few Central European finds, such as skull and dental remains from Goriac in Austria. In contrast, T. priscus was significantly larger and far more widespread, with discoveries ranging from the Iberian Peninsula to Germany and Hungary. The most important specimens were excavated from the Dinotherium sands at Eppelsheim. T. priscus is one of the earliest scientifically named fossil tapirs, introduced by Johann Jacob Kopp in 1833, the same year he also named the large species T. antiquus, though its distinct status remains debated. T. priscus disappeared around 8 million years ago at the start of the Upper Miocene, likely due to the Middle Valesian crisis, a period of cooling and increased seasonality. This left a fossil gap among large tapirs that was only filled about 2 million years later by T. avernensis, first described in 1828 from Les Étuaires in France's Auvergne region. Though rare, it was a consistent part of European faunas. Other smaller European tapirs from the Upper Miocene include T. balkanicus, defined in 1999 based on an upper jaw from Hrabarsko, Bulgaria. At the transition from the Miocene to the Pliocene, all small tapir species in western Eurasia went extinct and were replaced by medium to large forms. Although the genus Tapirus is thought to have originated in Asia, the first evidence of modern tapirs in the region dates only to the late Miocene, around 9.5 million years ago. Several skull fragments from the Upper Lushu Formation in the Linxia Basin, Gansu Province, northwestern China, are assigned to T. hessingensis, dated to about 8.3 million years ago. Around 2 million years later, lower jaw fragments from Shutangba, Jiaodong, in Yunnan Province, are attributed to T. yunnanensis. Both species were relatively small. In North America, the appearance of Tapirus is generally marked by T. johnsoni, the earliest known representative, dating back to the Middle Miocene. The type specimen, a lower jaw, was found in the Ash Hollow Formation in Nebraska. This rock unit, of Upper Miocene age, is famous for preserving entire skeletons of the rhinoceros teleoceras, likely killed during a volcanic eruption. T. johnsoni has a rich fossil record across central North America, already exhibiting all traits typical of modern tapirs. Some researchers also consider T. polkensis to be an early form of modern tapirs, although it's sometimes classified within Tapiravis. It primarily inhabited North America during the late Miocene. In the southeastern United States, particularly at the Gray Fossil Site in Tennessee, over 65 individuals have been documented, including complete skeletons. This relatively small species was about 15 to 20% smaller than today's lowland tapir. In the late Miocene, two larger forms, T. simpsoni and T. webbi, were present, the former in central and the latter in southeastern North America. T. webbi notably diverged from the classic tapir body plan with significantly elongated limbs. During the Pliocene, the number of tapir species increased globally. Some representatives from the late Miocene persisted in Europe, including T. arvernensis, a fairly common species in the evolutionary line of the Malayan tapir. Remarkable finds have been made at Camp dels Ninets in northeastern Spain, where at least 2 complete skeletons and several well-preserved skulls were recovered. Other species of this era include T. gianpivotoi and T. hungaricus, though the latter may be synonymous with T. arvernensis. By 2.7 million years ago, tapirs disappeared from Europe, likely a result of cooling climates, stronger seasonal variation, and expanding open landscapes. However, they survived in East and Southeast Asia. Compared to the Miocene's T. yunnanensis, the only known Pliocene representative, T. teilhardii, was significantly larger, although it is so far known only from a lower jaw and some isolated teeth in southern China. The Pleistocene fossil record is much richer. 2 evolutionary lineages have been proposed, one beginning with the relatively small T. pei, leading to T. sinensis and eventually T. augustus, also known as Megatapirus, the largest tapir ever, reaching the size of a horse. The other lineage is thought to include T. sanuanensis and lead to T. indicus, the modern Malayan tapir. Whether these lineages are truly distinct remains unclear. Aside from T. indicus and T. augustus, most Pleistocene tapir fossils come from East Asia and date to the early and middle Pleistocene, where they formed a key part of the Stegodonileuropida fauna. Notable finds include 2 partial skeletons of T. sanuanensis from Fancheng in China's Anhui province. T. indicus is the only tapir species outside the Americas to survive to modern times, although T. augustus may have persisted into the early Holocene. North America also boasts a rich Pliocene and Pleistocene tapir fossil record. Early on, Miocene species like T. polkensis and T. webbi were still present, later replaced by distinct species. Fossil evidence suggests that members of the genus Tapirus did not all appear simultaneously. For instance, in the southeastern U.S., T. lundoliaceae was replaced at the Pliocene-Pleistocene boundary by the massive T. haysii, which may have weighed up to 350 kilograms. T. lundoliaceae is known from Hiley Site 7C in Florida, while T. haysii has also been found in other layers of the Hiley Site, localities 16A and 21A, the Lisi Shell Pit, and along the Kissimmee River, all in Florida. The classic tapir of the eastern U.S. during the Pleistocene was T. varoensis, whose range extended from Florida westward to Oklahoma and Texas. A second distribution zone existed in the west, where the large T. merriami was eventually replaced by the smaller T. californicus. These two most recent North American species expanded their range northward, T. varoensis into modern-day Virginia and T. californicus into Oregon, indicating tolerance for colder climates. It's possible that tapirs persisted in North America until the arrival of the first humans. They only reached South America relatively late, following the formation of the Isthmus of Panama and the ensuing Great American Biotic Interchange. The earliest fossils from South America are around 2.5 million years old. Genetic studies support the idea that all South American tapirs descended from a single ancestral species. This is consistent with evidence that the lowland and mountain tapirs diverged only at the end of the Pliocene. The oldest known South American species might be T. gresselbini, known only from 2 molars found during drilling at Villa Ballester, Buenos Aires, Argentina. The exact age is uncertain, but associated fossils suggest a Pliocene context. During the Pleistocene, about half a dozen distinct tapir species evolved in South America, including T. rondoniensis, T. ryoplatensis, T. oliveraceae, T. terragensis, T. cristatellus, and T. mesopotamicus. Most are known from the late Pleistocene and likely occupied diverse habitats. For example, T. mesopotamicus was identified from a skull in the Arroyo Feliciano Formation in Argentina's Entre Rios province, while T. rondoniensis is based on a skull from the Rio Madeira Formation in Brazil's Rondônia state. Phylogenetic studies indicate that all these South American species form a monophyletic group and are more closely related to the lowland and mountain tapirs than to the Central American species. Tapirs have historically been forest dwellers, which made the widespread expansion of grasslands in the Neogene unfavorable for them. That, combined with the Quaternary extinction event, during which several tapir species vanished, has left us with only 4 surviving species today. That's it for now. In the next episode, we'll take a closer look at Kingler and its relationship with all insect Pokémon. Feel free to subscribe to our channel, leave a like, and check in next time.